know that these cancers are very aggressive, and the mortality rate is much higher. They account for 40% of the mortality, although they're very rare as compared to type 1 cancers. So these cancers are very chemoresistant, and so there's a lot of cell surface receptor targeting that's going on now. So what we did was, we've in the past, my lab has worked on using a tyrosine kinase inhibitor in uterine serous cancers, and they've shown that they work pretty well. There was a paper done in breast cancer where they saw that PI3 kinase, which is one of the downstream molecules in the pathway, once you inhibit PI3 kinase, that there's also some changes going on in BRCA. So we thought that maybe these two pathways are entirely different, like tyrosine kinase inhibitor is the ERBB2 pathway, and the Olaparib, the PARP, is a DNA damage pathway. So we thought that maybe there's some connection between the two pathways. So that's why we started investigating this particular thing. Then what we found out was, once you treat the cell with a PARP inhibitor, that it increases the HER2 expression on the surface. So that essentially provides more target to Neratinib, and the vice versa is true as well. We found out that when these cells were treated with Neratinib, that there was increase in PAR expression. Now PAR is basically the active part of PARP, so which means that there was more DNA damage done. And if you take away the PARP, which essentially helps with DNA damage repair, if you take that out and if you cause more damage, so then that's why the combination was very synergistic. So we found out that we did a mice study as well, and we found out that there was significantly a longer survival and statistically significant differences in tumor growth right from day 4 in one of our cell lines.